Thanks everyone for joining today's webinar. I'm Ibrahim Roshan. I work as a bioinformatician at EMBL-EBI. I'm part of the Protein Data Bank Europe group, which is in short form known as PDBe, where I mainly work on small molecule related data and services. I'm joined by my colleagues Preeti and Genevieve here. So without any further delay, let's get started. Today, we will be discussing about how to use PDBe KB Ligand Pages, a newly launched web page to explore ligands in the PDB efficiently. So before let me introduce to Protein Data Bank before I talk about the web pages. Protein Data Bank or PDB is a single repository of all experimentally determined 3D structures of macromolecules. PDB is managed by an organization called the Worldwide Protein Data Bank, and PDBe is one of its founding members. Structural biologists across the world deposit their structures to the PDB. These structures goes through a series of curation and annotation process, and before they get released to the public. So every structure that gets deposited to the PDB is assigned a unique PDB ID. A PDB entry consists of the 3D coordinates of the macromolecule. This macromolecule could be a protein or nucleic acids. Along with the macromolecule, there are ligands, ions, and water molecules attached to these structures. They will also be available as part of the PDB entry. And you can get complete details of this PDB entry by visiting the PDB entry pages, where you can visualize its 3D structures. You can go through the data qualities of the deposited structures. You can identify what are the ligands bound to the structures and get more details of this by visiting these PDB entry pages. Now, a PDB entry may correspond to the structure of a protein or nucleic acid. Now, PDB has structures of many proteins, and interestingly, in some cases, one protein can have multiple PDB structures. For example, shown here, this protein where the sequence is highlighted and the same color is used to highlight the part of the structure in PDB entry which maps to the protein. So we have almost 67 PDB structures mapping to this protein. And to make it easy to analyze all the structures related to a protein, we have developed a web page called PDBe KB Protein Pages. So this is just a snapshot of these PDBe KB protein pages, where you can assess the aggregated data of a single protein from multiple point of view. Now, the aggregation we have done in different levels. You can, in the structure, if you go and visit the structure tab, you could find all the PDB structures which are mapping to this particular protein. And in the ligands, you would identify all the ligands from the PDB entries which are mapping to this particular protein. And some cases, this protein would have some other macromolecular interaction partner. This could be another protein or a nucleic acid. So you could identify all those macromolecular partners by visiting the interactions. Now, there are other databases which annotate these proteins. So we aggregate these informations from all these resources and make available in a single place. And you can also identify similar proteins and their mapped PDB entries by visiting the similarity tab. Now, all the publications related to this particular protein and all the PDB entries which are mapping to this protein are also aggregated and made available in the publication section. Now, coming to the main topic of today's webinar, which are ligands. Now, PDB has more than 200,000 entries, and almost 76% of them contain one or more ligands. Some of the ligands are added as part of the experiment, acting as a crystallization agent. Some act as biological roles, like some of them act like cofactors, some are inhibitors, substrates, or drug-like molecules. Now, the complete definition of all the unique ligands present in the PDB are mainly given by 3 reference dictionaries called Chemical Component Dictionary, in a short form known as CCD, Peptide-like Reference Dictionary, or known as PRD, and Covalently Linked Components. Now, Chemical Component Dictionary contains the complete chemical description of all the standard and modified amino acids or nucleotides and small ligands, something like we have shown here in imatinib, and solvent molecules and ions. Peptide-like Reference Dictionary contains, as the name suggests, peptide-like inhibitors or antibiotics. Some of the oligosaccharides, like common oligosaccharides like mannose, glucose, sucrose, are also part of this dictionary. Every entry present in the Chemical Component Dictionary is called a chemical component. In some cases, PDB entries have ligands which are composed of covalently linked chemical components. Like in the example shown here, chromomycin, which has different carbohydrate chemical components and another CPH. So these are connected together. So we have consolidated all these chemically covalently linked chemical components in a single dictionary called covalently linked components. Now let's see what are the data and how it is represented in these dictionaries. So all the dictionaries, or each or three of them, every entry has a unique identifier, CCD ID, OPRD ID, or CLC ID. And you will have the systematic chemical names associated with these entries and the common descriptors like formula, smiles, inch key. Importantly, PDB is a structural database. It has the 3D coordinates of all these entries as well. For all the entries, you have three coordinates mainly. One is called a model coordinate, which is the 3D coordinates of the ligand atom selected from the first PDB entry this ligand was observed. Then there is an ideal coordinate, which is a computationally generated coordinate using softwares. Now, for each atoms, we have the bond connection between this atom, the type of bond, it will be a single bond or a double bond, and what type of stereotype is this bond. These information are also present in these reference dictionaries. In addition to these informations, at PDB, we also enrich these dictionaries with additional data using our own in-house developed tools. One of the tools we commonly use is a PDB CCD utils. You can parse this dictionary using this tool and generate more information for these small molecules, such as physicochemical properties, substructures, generate their 2D depiction image as shown here, create external mappings to other databases, find similar ligands associated with any of the ligands you are interested in. We use a tool called PDB RPGO to calculate protein ligand interactions from the entire PDB archive. We also have developed a tool called PDB Relic to identify functional annotation. As I mentioned, some of the ligands act as a crystallizing agent, some act as a cofactor, reactant, or drug-like molecule. So we use some heuristics to, based on the similarities, to identify what is the likely chance of one ligand acting like a cofactor or reactant. Now, how do you access all these informations related to ligand? That's where the PDBe KB Ligand Pages comes in. This is a single point resource to access the consolidated data of entire ligands in the PDB. This web page is distributed as mainly 6 sections, descriptions, which contains the common chemical descriptor names like formula, name, smiles. Physicochemical properties contains all the computed properties using RDKit. And we have aggregated all the PDB entries and proteins which are binding to the ligand in a single table in the section bound structures. We have also aggregated all the protein ligand interaction data, and we will see how it is represented in the later session. We also have collected all the ligands which have at least 60% similarity to the ligand in the related ligand section. Also, the ligand-specific database contains all the external mapping to different databases related to small molecules. Now, let's see how PDB ligand page looks like and how can you access these informations. So you can access PDB ligand page by visiting pdbe.org/chem. So I have here a video where we could see how we can access these informations. So you can search by name, formula, code. So here we will search for a drug called imatinib. It is a drug used against cancer and acts as an inhibitor of like tyrosine kinase. So its CCD ID, or it's the unique ID in the dictionary, is STI. So we will search using that ID. Once you search, it will navigate to that particular PDB KB ligand page for STI. So it has those 6 sections: description, physicochemical properties, bound structures, and interactions, aggregated interaction information, related ligands, and the ligand-specific databases, which has all the external mappings. In the coming slides, we will visit each of these sections and see what type of data is present in them. So going to the description section, you can access the chemical descriptions like name, smile, inch key, all those informations. And there is a copy button given there, so you can click that to copy all those data easily. And there is a field called first observed in. this is the PDB entry from which the definition of the ligand was created. So as I mentioned earlier, every dictionary has the three model coordinates and ideal coordinates. Now the model coordinates comes from the first PDB entry in which this ligand was observed. Now there is an image gallery adjacent in the description section where you can identify, once you see the video, it might be clear. You can go and navigate to the image gallery to see what are the different fragments and scaffolds associated with the ligand. So we pre-calculate these fragments and you can easily identify what kind of fragments are present in this particular ligand. You can copy the ligands, fragments, smiles, inch-key, these descriptors and search against any other databases. Now coming to the one important aspect of description section is the view. You can view the ligand in the 3D coordinates as well. So you go to click the view 3D button, it will pop up the 3D viewer where you can select either the ideal coordinate or the model coordinates of the ligand. And there is another field where you can select corresponding to the 2D image gallery. You can see the different highlighted views of the fragments and scaffolds in 3D view as well. You can swap that for each ideal and model coordinate, see how they vary and what kind of confirmation they take in. You can also download this ligand in different formats from the download as button. Now coming to the next section, which has the physicochemical properties. So these properties are all computationally calculated properties. We have calculated them using the RDKit. So this gives you a quick overview of the main properties of the ligand, if they are following what kind of properties are there, like how many atoms are there, how many heavy atoms are there. These informations are grouped together for you to identify and quick overview of how the ligand's properties looks like. You can also download the entire list of the properties by clicking the download properties button. This will download as a JSON file with each field and their values. Now the next section is called bound structures table. So this is a table where we have aggregated all the proteins and the PDB structures the ligand binds to. So this is located just below the physicochemical properties, and this is shown as a table where you could find the protein's name, the UniProt ID associated with that protein. And we have also given how many proteins and the PDB entries this ligand binds to. It also has the ligand function column, which shows what kind of ligand function annotation might be present in this. In this case, we know STA is a drug. So since these are computationally driven, so we have given drug-like properties. And once you click on so I will go back again. So once you click on the protein UniProt ID, it will go to the PDBe KB protein pages, where you will get all the aggregated information related to that protein. For example, you can find the entries or PDB structures associated with that protein. You can also visit the other ligands which are binding to this particular protein here. And all these ligands also have some kind of annotation. You see the drug-like annotation there. So these are other drug-like molecules which are binding to the same protein. So it would be interesting to see if they have any kind of structure similarity. Once you click on that, it will go to its own ligand page and describing all the properties related to that particular ligand. So here you could navigate between different pages to know more about each different ligands and proteins. It also has a structure view. So once you click on that, it will show all the PDB entries in which the ligand binds. So once you click on the PDB entry link, it will visit the PDB entry page. It will show all the ligand PDB structure interactions, what type of residues are interacting, what type of interactions are there, what is the distance between these two atoms in the 3D coordinates between this atom, how they are interacting. These informations are also available there. You can easily download the entire information as a CSV. You can download the table as a CSV by download structure CSV. And if you want to get the entire PDB entries information, you can download them as a coordinate MMZip file. So which is the 3D structure information of all the PDB entries. Now coming to the next section is the interaction statistics. Now the earlier session we show what are the entries and proteins they are binding to. Now we aggregate these binding interaction properties in a very interactive way. So this is just below the bound structure table. So here you have a interactive view of a heat map component and a 2D display of the ligand with their interaction statistics shown here. So the green shows the strength of each atom's interaction as compared to the total number of interactions. So here the green color is depicted both in the heat map and the 2D display. In the 2D display, you can see the size of the circle and the intensity of the color indicates the value. So the darker and the bigger the circle means that atom is mostly interacting as compared to total number of atoms interacting in the entire ligand. So you could identify what are the main fragments in a ligand interacting from the entire archive. So this gives you an idea about how the ligand might be interacting with the other macromolecules. So let's see how you can navigate this information. So if you if you want to know more about which atoms are this interacting, you have this slider button. This has all the ligand instances information, like so how many structure this data is coming from, how many protein this data is coming from. So if you drag the slider, it will show the ligand atoms names. So at a time, it can only show 25 atoms. So if you zoom into that, it will show which ligand atom is interacting. Once you mouse over, it will also highlight in both the places. If you mouse over the 2D display, it will highlight in the heat map component. Now, coming to the next part, you have another information, which is the interaction strength between a ligand atom and an amino acid. So this is calculated as a ratio between the number of interaction between a particular ligand atom and a particular amino acid with respect to the total number of interaction that amino acid is having. So this is kind of metric of like how likely to a ligand atom might interact with an amino acid. So this gives you an insight about what kind of properties are this ligand having, what kind of properties this ligand might be binding in a binding site. So this gives you a quick overview of how the ligand is acting in the entire PDB archive. So yeah, so these are the values. So and you can sort these values based on the amino acid properties, whether amino acid like hydrophobic or positively charged, or also you can sort based on the interaction value. And you can also filter this interaction based on the interaction type. If you're only interested in hydrogen bond, the values gets updated by selecting that. So it's a multi-select button. You can add as many interactions you would like to see, and all the values gets updated accordingly. You can download all the interactions by downloading the download interaction button. And as I mentioned, the earlier PDB RPG is the tool we use to calculate the interactions. So these are the equations, as I mentioned earlier. You can visit them later, like what these values mean. Now, the next section is the related ligands section. It identifies all the similar ligands or the ligands with the same scaffold, or even the ligands with which are like stereoisomers of the ligands. So this is located just below the interaction status section. So here we have shown MPZ, which is the ligand with the same scaffold. And we show here similar ligand section, we only show ligands which have at least 60% similarity. And once you I will just go back so that it's clear. So for each ligand, we have given how many PDB entries it has bound to. In this case, this is bound to one PDB entry. So once you click on that, it will visit the PDB entry page, which is corresponding to which PDB entry it is binding. Once you visit there, you will see the ligand information there. And as I shown earlier, if you click on that, you will find the interaction between that ligand and a particular entry. And in the similar ligand section, same fields are available. You can visit and also you can filter them. So if you give 70% similarity, it will show only those ligands which are having similarity more than 70. And each ligand has its own clickable link, which will visit to its own ligand page, where you would get more information about that particular ligand. So here, every information is connected together so that you can navigate between these informations and compare and match and try to answer a different scientific question as per your interest. Now, the last section in the page is the ligand-specific databases. So once you know, so these are the information in the PDB. Now you might want to connect this information and collect or get more information from other databases. So we have collected all the linkages to other databases like EMBL, where you could get the bioactivity data, drug bank, where you could get the clinically approved drug target informations. So these are the links available. And now if you want to do a batch process analysis, you can also download those informations as a CSV file here by clicking on the download or database IDs information as well. Now, while we were developing this PDB Ligand Pages, we also collaborated with the Cambridge Crystallography Data Center, or known as CCDC, who manages the Cambridge Structural Database, or CSD, containing over 1.3 million experimentally determined 3D crystal structures of small molecules, and also collaborate with the EMBL. They have the bioactivity data to integrate the structural. The PDB also has structural information, CSD has also structural information, functional, like EMBL has the target information, and biochemical annotations of small molecules and their target from these three resources. As part of this collaborative effort, we were able to identify common ligands present in these resources. So this image shows the number of ligands we could identify. So this mapping was done based on the complete mapping of the ligands. There could be ligands which are similar, missing it, but for the time being, we have only mapped which are the exact mapping. So almost there are 3,000 ligands we could find in these three resources. So we could add more annotation, get more information that are already existing in these resources to ask more questions about these ligands. And we recently released the bioactivity data for almost 17,000 experimentally determined protein ligand complexes. So by getting the intersection of PDBe and EMBL. So the data table somewhere looks like this. So for each PDB entry, which ligand it is binding, which chain ID is, which chain it is belonging to, and what is the protein uniprot ID, and what is the target in the EMBL, and what is the bioactivity value in it. So this is just a snapshot of the bioactivity data. You can know more about this bioactivity by clicking on the bio report. You could download this file and analyze what data present in it. Now, to just recap what we have discussed so far. So as a user, if you want to know about a PDB structure, you can go and visit the PDB entry pages. And like if you are interested in a protein and want to know more about the different PDB structure belonging to that protein, you could visit the PDB KB protein pages. Or if you want to know more about a particular ligand, you can go and visit the PDB KB Ligand Pages. Any page you visit, you could visit any other pages. There are any connection between these entities, then they are connected together, which helps you to ask and create questions and find more interesting aspects and aspect of any of protein or PDB structure or ligands. Now, to know more about and what are the resources available, you could visit any of these links. We make available all the data we generate through FTP freely. So those files we use to create this data are available in the FTP. Also in the web pages, we use the APIs to get the data. So you could also use for your own analysis these APIs to query against the database we have available. Now, how to use like the integration of PDB and EMBL has provided, you can get more information about the bioactivity data, compare them against the interaction data. So there are some training material we have created. You could visit them and get to know how you can also do for a protein or ligand of your own interest. So that sums up my presentation. So if you have any, I hope you guys have a good idea of what PDB KB and PDB entry and PDB KB Ligand Pages are there. So if you have any question, please feel free to ask. Thank you.
Right. Thank you very much, Roshan, for your presentation today. And thanks everyone for joining in and sending in your questions. We are also joined by other panelists who are from PDBe. They are helping with the Q&A. All right, so we have a few questions already in the Q&A box. Read the first question here. So one question here is about the PubChem submissions. Who makes your PubChem submission and what criteria are these submissions are selected from your side?
Related to submission, maybe Genevieve, can you? I'm not sure what PubChem was mentioned, but maybe Genevieve can give some idea about.
So we do sync with other databases. We've just been presenting how we sync internally to Campbell via there's a Unichem connection, and there was a nice slide that showed that as well. The PubChem submissions, I'm afraid I'm not quite sure on the exact mechanics of our links appearing on PubChem. I will have to ask a question and get back to you on that.
There's another question. Are the criteria for other database matches based on ING key exact match?
Yeah, that's correct. So we are mapping based on exact ING keys. And we are using Unichem to establish the mapping between various databases.
Are you able to search the ligand database if there are fragments or small molecules from in silico screens?
So just to clarify, the ligands that are available on the PDBe KB pages are ligands that have been observed in an experimental structure that's been deposited. So these are experimentally observed small molecules. So that being said, there is different ways in which we're highlighting on these pages scaffolds that are found within those ligands, and that may or may not be useful for you. Yeah, but these are not docked ligands that have happened in silico. These are ligands that are occurring and present in those 200,000 structures that are present in the database.
Is there any database for nanoparticles acting as ligands?
So the PDB itself is for experimentally determined macromolecules. And if something that was, I'm trying to think what a nanoparticle chemical or ligand is. If that exists, if that, for example, if the nanoparticle was somehow generated by RNA or DNA, like I know there's some incredibly interesting things that people build out of DNA and RNA. I don't know if they're quite nanoparticles, and that could end up in our database. But yeah, it's depending on what scientists are doing, whether they'll be present on our database or not. If you wanted to look into nanoparticles, it's probably be good to understand. The composition would be important to understand in terms of how best to search for it. Because if it's composed of nucleotides, it might be that you want to search like a DNA or RNA. You might want to search a DNA or RNA sequence in order to find it, rather than necessarily looking at ligands.
Okay, so the next question is, I think, about the database curation. If I have published a paper about ligand-protein interaction, will PDB upload it on their site? And if yes, how can I find it?
So those interactions we have shown here, those are computationally calculated. So these are not particularly what is reported in the paper. So at the moment, I'm not sure if they are like currently in the PDB entry pages, they are not available, even if some interaction is reported by the crystallographer. Let's say this is observed, that is in the paper only. But all the calculations, all the statistics we show, these are like we do the calculation based on the distances between the atom, based on the 3D coordinates. These are all computationally calculated. But there should be if something is observed in the 3D coordinates and reported in the paper, there should be a overlap between what those two observed. May not be the exact match, but because of the maybe some randomness or some calculation differences in the viewer or the software used. But to be clear, like all the information are computationally calculated by using the tool PDBe RPGO and not from something reported in the paper.
But there are publications on the tools that we use in order to do the calculations. Like, so Arpeggio has a publication, and I think, are those links available on the pages themselves?
So we have the link to the GitHub. The GitHub has the information about the tool. So this PDBe RPGO is a refactored version of already existing tool called RPGO. So we have made it a Python installable and pip installable tool. And you could use this tool for your own analysis, or even after docking study analysis, you could calculate the interaction between a macromolecule and a ligand using this tool.
All right. So next one is, can we load the structures in Chimera software and do the remodeling based on ligands as needed?
Yeah, I mean, we have the files available. These files are supported by software. So this question is particularly related to the Chimera software. So I can't really talk more about what are the supported in that. But most of the files supported by all the files which are supported by different softwares, we are making it available in our web pages. So you could download those files and then use your own software of interest to do analysis, modeling, things like that.
Maybe a helpful thing to distinguish is all experimental structures have unique PDB identifiers. All ligands have unique identifiers as well. But if you fetch a PDB entry based on its identifier, all of the ligands come with it, with the unique identifier embedded in that file. I don't know if that helps. And it's that unique identifier of the ligand itself that is what's the key for these ligand pages in order to find them.
Right. So the next question, I think if I can rephrase. So is there information available on this page about protein ligand docking analysis so that a person can find where ligand is binding exactly to what residue in protein structure?
Yeah, we do have that information. So based on the calculate interactions, we show, I think one of the videos I showed, if you go to the PDB entry page, for each ligand, we have the display of ligand interacting residues informations. So you could match that to identify the grid possibly. So we calculate this based on like finding a particular, like 5 Armstrong 3D volume around the ligand. So that would it will only identify if the interacting atoms are of like the complementary type, but some of those interacting atoms will be in that that range. So you could use that to just create a grid around that ligand. So yeah, it will help you to do that. So these are available from the entry pages.
Yeah, so just to also add to that, if you from the ligand pages, and Roshan showed this, there's like this table of the proteins and structures that's central within the ligand page. Or there's links on that will take you to individual entry pages, and I think it lands specifically on the interaction part of that page, which focuses in on looking at the ligand and where it's positioned within the structure and what interactions are occurring. Yeah, so it's a table that has, I think, that bound structures and bound proteins, and you can flip between those two options, the two radio buttons, protein and structure.
There's one open question here. I don't know if I can rephrase it, but are there like disease-related information available on your ligand pages? People can screen for if they are studying any specific disease.
So, I mean, one of the really cool things about the way our pages work is they're linked to the UniProt classification system. And in UniProt, proteins will be classified often based on disease. You can do a search based on disease and find all the UniProt identifiers. You can then use those UniProt identifiers to go through the PDBe KB pages, which are protein-centric, and you can find all examples of the experimental structures that have been determined for that set of proteins that belong to that particular disease. So that might be a helpful way of investigating. Prudy can speak more to this. I think we do have disease highlighted on the KB pages, landing pages for proteins themselves as well.
Any estimates of the count of authentic ligands in pockets rather than just any old hetero compounds?
There are some quite cool tools for looking at pockets that are available from the PDBe KB pages. So there's a ligand subpage on the PDBe KB pages where you can open up a view and see essentially an overlay of all of the instances of a particular protein in the PDB, so a particular chain of protein, and then all of the ligands that have been seen bound to it. And if you know something about like what are common crystallization ones, you can turn off the ligands that are common crystallization ones and just look at the ones that are the interesting chemicals. So that's the not the KB Ligand Pages, but the PDBe KB Pages. And that can be sometimes quite a helpful way to, if you want to can think about the ligands in the context of the protein, is to go to the protein pages, which again, that central table where you've got the links to every instant, you've got links out. If you have the protein radio label selected, I think, and you click on one of the UniProt IDs, that will take you to a KB page, and you can then go to the ligand page in the KB page, and that will, it's like we're coming at it two ways. We're coming at it ligand focused and then spanning out, and then coming protein focused and then looking at the protein from a ligand perspective. So yeah, the ligand pages on PDBe KB can be really helpful. And PDBe KB, it's pdbekb.org will get you there. That's also quite an easy one to find.
Thank you. Could I change the protonation state of the ligand and investigate how that is affecting the ligand-protein interaction?
Yeah, yes. So you could use the tool PDB Arpeggio. So this is like an open source tool. So so it does not do any. This tool does not do any protonation steps and things like so. So you would assume the input to the Arpeggio is something protonated, processed with all the problems problems. So PDB Arpeggio helps you to calculate the distance between atoms and find a different type of interactions. So you could use your preferred way of protonating. So there are different type of softwares using different methods. So you could try any of these methods, create a file, to use that file to PDB Arpeggio to calculate the interactions. Yeah. So if the protonation changes, the interaction properties might also change. Yeah. Yeah. So the answer is like you could use that.
It's unfortunately an offline tool. We don't have it embedded in the pages, so you would have to download and use it, but it is possible.